is Dr. Almeida-Porada, and she's from Winston-Salem, and she's going to tell us about cellular therapy for intestinal regeneration.
I'll just put it in my notes. That's okay, thanks. Good morning. First of all, I would like to thank Aubrey and the SENS Foundation for inviting me to this meeting. And I'm going to talk about cellular therapies for intestinal regeneration in the context of IBD or inflammatory bowel disease. So IBD is defined by inflammation of the gastrointestinal tract in the presence of uncontrolled immune response. And there are several approaches, drugs that have been used to treat this disease, but although these drugs will induce remission, thus far we don't have efficacious treatment for this disease that will lead to a cure. IBD represents a significant and rapidly growing healthcare burden in developed countries and impacts the life of millions of individuals. There's more than 1.5 million people in the US and 2.2 million in Europe that suffer from IBD. IBD is a bimodal disease and can occur later in life, around when people reach 50 or 60 years old of age, but really can begin at any time. And there's another peak that occurs in teenagers and young adults, and actually is the biggest proportion of patients the onset of the disease will be in early years. Therefore, will lead to a lifetime of care. And statistics published by the American College of Gastroenterology show that this burden is for the patient and for the healthcare system leads to around 2.3 physician visits and costs around $3.3 billion per year. So we know that genetic factors make a major contribution to the pathogenesis of IBD, but not only those are important. We know that environmental and development factors also have been shown to impact the outcome. For instance, people that live in the countryside and move to a more industrialized area, for instance, the city, they are more prone to develop IBD than people that will remain in the countryside. And we know that abnormalities of the innate and adaptive immune system are responsible for the balloon and controlled immune response to the gut flora. So it's really the immune dysfunction that is at play in these patients. And we know that in Crohn's disease, there's a skewed Th1 and Th17 cell response with the production of gamma interferon and IL-17, while in ulcerative colitis, the immune response is Th2 mediated with overproduction of IL-4. But it doesn't matter, it's the skewed in immune function with alteration of immune function that leads to the end result, that is a poor regulated T helper response, decreased in T cell apoptosis. There's increased numbers of local dendritic cells. There's recruiting of cellular inflammatory infiltrates that will lead to the ulcerations of the mucosa and inappropriate or delaying of healing. But not just the immune system is at play. We know that there's severe cellular dysfunction in these patients, and alterations in the stromal or mesenchymal cells and myofibroblasts have a very essential role in this disease and will perpetuate intestinal inflammation and will contribute to the fibrosis. Also, the microvascular and endothelial cell dysfunction will lead to persistent tissue hypoperfusion, ischemia, and therefore will contribute to chronic inflammation. And what is curious is that in a lot of these patients has been reported that despite this pro-inflammatory condition that would lead to increased numbers of endothelial progenitor cells in circulation, several studies have pointed out that this population is decreased in the peripheral blood. So people have thought about a way of curing this disease, not just treating or ameliorating, but curing. And because of the heavily cellular dysfunction, we thought that biological-based therapy would be a more appropriate approach for this disease. If we think about a biological approach, we have to have cells that can, of course, decrease the inflammation and apoptosis, that can reset the immune system through immunomodulation, can normalize vascularization, will induce regeneration of endogenous cells, and therefore replace the ailing or destroyed tissue. It contributes to tissue-specific stem and progenitor cells. So this would be the ideal cell therapy to treat and achieve a cure for these diseases. Mesenchymal stem cells have been used and are ideal for cell therapy since they don't seem to have side effects, and they have been used in graft-versus-host disease quite successfully because they play a role in immunomodulating the immune system. And so there's been clinical trials using these cells at the concentration that were used for graft-versus-host disease. And the use of these cells is because they have the ability to migrate upon infusion to areas of inflammation and injury. And they deliver actually several factors that can promote healing, not just modulating the immune activity, but they are able to inhibit apoptosis, and they can stimulate the resident or progenitor cells. So why is that we were not successful in using this therapy for IBD? Only around 50% of the patients respond successfully to this therapy. And looking at what happened, two things could have happened. One is that the mesenchymal stem cells indeed are able to modulate the immune system and dampen the immune response, but in situ they are not able to decrease inflammation because not enough cells can reach the intestine to cause local decrease of inflammation. And the other factor is that it has not ever been reported whether these cells can indeed contribute to the stem cell niche in the intestine to cause revascularization and cause the intestinal stem cells to start proliferating and cause a normal phenotype. In these studies that I'm going to present, we went the next step. Another cell type that is very important and has been used for other situations in which vascularization was needed are the so-called endothelial progenitor cells or endothelial colony-forming cells, because we know that these cells can promote angiogenesis via paracrine effects. They can cause endothelium remodeling, and they have vessel-forming ability. And although they have been used for infarct and ischemia in the peripheral, in the limb system, they have never been used in a situation of IBD. So the aim of these studies was to investigate the contribution of mesenchymal stem cells and endothelial progenitor cells to the intestinal vasculogenesis, stem cell pool, and cellular composition. And we wanted to go the extra step and identify a subpopulation of mesenchymal stem cells that maybe could have enhanced intestinal engraftment capability so that we could get more cells to the site of injury and therefore cause an effect. And also we wanted to see whether these mesenchymal stem cells could really contribute to the stem cell pool. So instead of going with, or for this first part of the studies, with the injury model or IBD model, we decided to go with the physiological relevant model in which we could study homing of the cells to the intestine, because we know that mesenchymal stem cells will home to a site of injury. Therefore, we wouldn't have a correct answer if we were looking at populations of cells that migrated to the intestine. So for many years, we have been using the sheep model for human stem cell activity, in which we know that we can engraft cells and follow these cells through the life of the animal. We transplant these cells in utero at a time that the immune system does not recognize the cells as foreigner. So they will allow the development of tolerance and long-term engraftment of the human cells. The cells persist in the animal and is a competitive engraftment because there's a normal physiological environment. Therefore, all the cells are proliferating and giving rise to the organs. So we have a good stimuli for donor cell differentiation. But of course, human cells are going to have to compete with the normal engraftment of the animal. So we were hoping to see if the cells migrated, or if there was a population of cells that migrated more to the intestine, and then see the effect of differentiation of these cells into intestinal stem cells. And the way we do it, we just do an ultrasound injection at 45 or 65 days of gestation into the sheep fetus. Here you can see the liver, and just for purposes of showing, this is actually an intrahepatic injection. We usually go into the intraperitoneal cavity, but you can see the injection is done the same way. You can see the needle coming into these are the rib around the liver. This is the liver, and then the needle is placed inside either the liver if it's an intrahepatic injection, or next to the intestines in the intraperitoneal cavity. We remove the needle as we inject the cells, and then we check for the heartbeat to show that the animals are still alive, and we don't lose really any animals after this procedure. So we use our cell populations. We use endothelial progenitor cells and mesenchymal stem cells. The endothelial progenitor cells, the following phenotype, they didn't have markers of, or very low markers of CD34 or hematopoietic stem cells. They had 105, CD31, a little bit of 133, low 117, and these cells were actually provided by Dr. Yoder at Indiana University. The mesenchymal stem cells that we used clonal cells because since we are trying to address the question which cells would migrate to the intestine, we did single cell deposition of bone marrow stromal cells isolate based on STRO1 and 45 negativity. We visualized the cells in the wells and confirmed that they were single cells, expanded the cells for 3 months until we had enough for the transplant. And we know that most of the clones were expressing at high levels CD90, 29, and 105. They express at low levels 31, 51, and 102. And they were completely negative for CD34, 38, and 133. So we transplanted 8 different clones at 1 million cells per fetus. That gives around 10 to the 7 per kilo. And at 2 to 3 months post-transplant, we evaluated the intestine for the presence of human cells by in situ hybridization. And in some of the studies, we transduced these cells with GFP, or in case of the endothelial cells, with red fluorescence protein, so that we could better track these cells upon transplantation. So we first looked at the cells by in situ hybridization. And here you can see normal control cells. This is stained with cytokeratin and the human-specific probe. This is a human intestine control, showing that the probe hybridizes to the human cells. And here you can see an example of one of the animals in which we saw engraftment mostly into the mucosa of the intestine of these animals. And here you can see the percentage of engraftment in the different clones. And you can see that in 2 of the clones, they had around overall 5% engraftment in the mucosal area of the intestines. So then we looked at the intestinal stem cell area as defined by Musashi, that's a marker for intestinal stem cells, still looking at the human-specific probe. And above you can see the control, the human intestine, and here you can see that the same 2 clonal populations that had increased engraftment, overall engraftment to the mucosa of the intestine, had a higher contribution to the ISC, or the intestinal stem cell pool, giving rise to levels of 15% of cells, human cells, within the intestines of these animals. Then we looked for other markers to further confirm that these cells were indeed intestinal stem cells, and LAGR5 is one of the markers for intestinal stem cells. And here on the right, you can see that the cells that had the human probe, and they were Musashi, that is a marker for stem cells, intestinal stem cells, and LAGR5, they are also positive for LAGR5, confirming that these cells were contributing to the ICC, the intestinal stem cell area of the intestine. So we start looking what was special about those 2 clones that caused enhanced engraftment to the intestine, and we did proteomics and genomic analysis, and we come up with a series of markers that were putative markers for our cells. And amongst those markers, we were looking for a marker that then in the future would be a surface marker that would allow us to sort those cells specifically to use in a clinic. So all those markers that we identified, EphB2 was the only one that was a surface marker that would allow us in the future to sort these cells out amongst the mesenchymal stem cell populations. And indeed, at the bottom column here, row, you can see that one of the clones that gave rise to more cells was expressing EphB2 at higher level than the other clones. And then when we looked at just stromal layers or mesenchymal stem cell layers, you could see that certain cells within the stromal layers were EphB2 positive, while others did not express EphB2. So we knew that we thought that we had found a marker that could pull out cells that would increase engraftment to the intestine. And the next question that we asked, it was whether if these cells were indeed intestinal stem cells, they could, in an explant culture, to give rise to crypt-like structures. And we asked the question whether these crypt-like structures would be also EphB2 positive, and EphB2 is known to be present at the intestinal stem cell niche in intestine. But we harvested from one of the animals that had been transplanted with the high clone, high engrafting clone. We did explant cultures, and we could see that by day 21, they gave rise to crypt-like structures in vitro, and these cells were Musashi positive and EphB2 positive. So the next experiment, it was to isolate now these cells using an antibody using EphB2 and see, comparing with EphB2 low, and see if we still obtain the same effect. So we did that. We sorted EphB2 high and low and labeled the cells with GFP, and then looked at the engraftment in the animals. And we found that EphB2 positive cells, in similarity to the clonal studies, gave rise to 5 or 6 times more engraftment into the intestine than the EphB negative counterpart. And here you can see the cells with the GFP expressing lager and Musashi and EphB2 engrafting in the intestine of these animals. We also asked the question that whether upon engraftment, if the cells would start proliferating in a mucosa. And we use Ki67 as a marker of proliferation, and you can see the cells coming and proliferating in a villus of the intestine, giving rise to more to cells. Then we did similar experiments using endothelial progenitor cells. We transplanted the cells at different doses. And at the end of gestation, we collected intestines and from the animals that were transplanted and looked. The endothelial progenitor cells had been transduced with red fluorescence protein so that we could track them better. So we did fish and look at and also tracked through the DsRed protein. And as you can see here, where we have the intestine divided in sections, the EPCs engrafted at high levels in areas also of the crypt of the lubricant, so in the stem cell region. And here you can see the different animals that were transplanted. And they constituted 23% of the cells in the crypt of the lubricant, and actually 81% of the cells were transplanted located in that area. The overall engraftment of the intestine is shown by the blue bars, and it was a little bit lower, but really in a stem cell region, the engraftment was quite high with one of the cells that had, one of the animals that had a higher dose reaching 35% of cells in that area. To confirm that what we were looking, it was the cells that had been transduced, we looked at the DsRed population, and then we stained with an antibody, in this green antibody, or FITC, against DsRed. And here you can see the colocalization of the DsRed and the DsRed antibody and the green antibody, showing that the cells were indeed the cells that we had transplanted, and it was not just an artifact. Then we start looking at the contribution to other cellular subtypes in the intestine. And as I said, myofibroblasts are important population that needs to be replaced in IBD. So we looked at smooth muscle actin and vimentin, and we found that cells that had engrafted in an interstitial area of the intestine were contributing to this population, and we had engraftment level at around 28% of myofibroblasts. If you recall, in one of my first slides, these cells were expressing CD133, but at low levels, and upon engraftment, 73% of the cells start expressing or further express CD133, and CD133 is also a marker of intestinal cells, and they know that there's a role in cancer. So of course we can say here that they are differentiating into a population of cells since they had low levels of CD133, but these cells are upregulated and they are situated in a vasculature area of the intestine. They also express intestinal stem cell marker that they didn't express prior to transplant, Musashi. And you can see several Musashi positive cells in the intestine. It was around 10% of the contribution to the intestinal stem cell. A marker of ICCs, or the cells of Cajal in the intestine that provide control of the motility, express CD117, and these cells also we found that engrafted and they are expressing CD117. Actually 99.5% of human cells were CD117, and they composed 8.1% of the total CD117 cells, intestinal cells that we found. in these animals. We didn't see differentiation, or very few differentiation, into enteroendocrine cells as determined by chromogranulin A. And by contrast to the mesenchymal stem cells, that a good proportion differentiated into cytokeratin positive cells, the endothelial progenitor cells remain at the base of the crypt, and they didn't contribute much to the cytokeratin population. And of course, endothelial cells shed particles, so we were curious whether this massive engraftment that we found with endothelial progenitor cells was caused really because these cells engrafted, or it was particles that were shedding. So we looked at the cells that we hybridized with a human sheep-specific probe, and looked if the cells that were expressing the DsRed, here in green because we used an antibody against it, were expressing sheep DNA, and none of the cells are expressing sheep DNA, showing that these are the cells that we transplanted and not just particles that were released and integrated by the cells in the sheep intestine. So in summary, EPCs engraft at eye level in and around the crypt of Libercan region. The EPCs contribute to the ICC population. EPCs contribute to the stem cell pool, and they differentiate into intestinal cells at low levels, and they contribute to less than 1% to the enteroendocrine population. By contrast, mesenchymal stem cells that are high for ephrin, they engraft also at high levels in the crypt of Libercan. They contribute to cytokeratin positive cells. They repopulate the intestinal stem cell pool, giving rise to Musashi and Lager cell positives. The explant cultures are able to provide a crypt-like structure, and as I said, they generate cytokeratin differentiated cells. So we have identified a marker that allows the discrimination and isolation of a novel subpopulation of mesenchymal stem cells that has higher engraftment capabilities, and therefore we hope now in an injury model that we can time these cells so that we can treat better the injuries or the disease of the bowel. And also the endothelial progenitor cells constitute a novel and promising therapy for replenishment of the crypt of Libercan region, the ICC compartment, and the myofibroblastic population. So in the next step of studies, we are hoping to use IBD model, murine model, in which we are going to time a mesenchymal stem cell and EPC therapy to see what is the outcome in these animals if we really can provide a successful cellular therapy for these animals. And just to acknowledge my collaborators, this study was started at the University of Nevada while I was prior to move to Wake Forest. And the people in my lab that work in this project, it was Evan, Colletti, Josh Wood, Takashi, and Jason Chamberlain, my collaborator Christopher Porada, Dr. Yoder and David that sent us the EPCs, and Dr. Zanjani as well at University of Nevada. Thank you.
Very much. Are there questions? My question is, does this work in the mouse model, and does this work in an adult mouse? Because this is a sort of embryonic sheep model, and ultimately you're going to want to test this in an injury model where there is a concern, at least why I'd be worried whether the ulcerated regions in the IBD situation, would they actually allow any engraftment at all?
Exactly, so that's what we are trying to do. I mean, we have not done those studies, so it was proof of principle that we couldn't isolate the population in injury model. So we had to go with a normal model to isolate the population that would engraft better in the intestine. What we are trying to do now is to time mesenchymal stem cells with different phenotypes. So the idea is going mesenchymal stem cells that will reduce the immune response and decrease the inflammation. and then give different injections in which we'll then start replacing the intestinal stem cell region with other cells. So you have a good point. We have first to reduce inflammation and promote an environment that is suitable for engraftment of the other cells. So if we are hoping that if we combine EPCs and mesenchymal stem cells, several injections, that we can reduce inflammation and then promote vascularization and then replenishment of intestinal mucosa. But of course, we have to do the next set of studies with the injury model, and we didn't get there yet. So.
Well, if there are no more questions, I suggest we move on to the next talk to sort of get approximately back on time.